All right, so we completed part one, which again, as I said before, is examining the gap that exists between inanimate matter and animate matter. And now we move on to the shore of life and examining now the fundamental unit of all life, which is of course the cell. And please remember that at the time when the theory of evolution was framed, the cell was a black box. They did not understand how it functioned. In fact, they called it a bag filled with protoplasm. It was something that was almost gross, and it's not their fault because they did not have access to that information. But now what happens is that that black box has been opened, and then we understand more and more its inner workings and its intricacies. And so what we're going to do is to step on that shore of life and to examine what we learn from the cell, especially in regards to this important questions that we are looking at today. So essentially, while unicellular organisms are more simple, the cell, it is anything but simple. And some of you might not have a training in biology or cell biology, but that's okay because in fact, there's a beautiful analogy that exists between the body, higher organisms like you and me, and the cell itself. So essentially, we have a head where the information is processed and contained, and so the cell also has a head, a nucleus, where the DNA, where all of the information is contained and processed. And we have skin that basically protects us from the outer environment and allows us to interact with this environment along with the different senses. And the cell also has a skin, which we call the cytoplasmic membrane, through which it interacts with its environment. We have a whole bunch of different organs inside our body that serve very specific purposes for the body, and we discovered that inside the cell we also have all of these organelles that have specific functions. So as we go through this part, you'll see that a lot of these things apply not just at the cellular level, but it also applies at the higher organism level. Now, when we do things in our environment, the way we relate to each other, the way that we do different things, we use our hands, we use our mouth, our eyes to see, our ears to hear. But what about the cell? How is it that the cell is able to do different things? It's a very different world at that microscopic level. And so essentially, a cell is a complex association of various molecular structures, proteins, nucleic acids, fatty acids, phosphotriglycerides, etc., etc. And all of these lead to the ability of the cell to carry out various complex molecular functions: metabolism, replication, signaling. We talked about photosynthesis also earlier on. And we could talk a long time about all of these different structures and processes, but I'm just going to pick one, which is the enzyme. The enzyme is a huge molecule. There are so many different types of enzymes that are able to do different type of work. When I was working in the lab and my chemistry didn't work out, I didn't get the product that I was hoping to get, and my boss would say, well, what happened there? I said, well, wait, I don't hold the molecules by the hand. They do what they do. Well, where we fail as chemists, these enzymes are incredibly powerful. They're able to take molecules and put them together, break them apart, do all kinds of modifications that make us chemists jealous. We wish we were able to do this. And when the structure of the enzyme was discovered, we said this is a huge structure. Just to give you a little idea as to how big this structure is, the red dot that you see on the screen would be about the size of a water molecule. So you can imagine how big this molecule is compared to a water molecule. This is gigantic. It's a huge three-dimensional structure. And so chemists right away and biologists wondered, why is it so big? Why is it so huge? It seems like such a waste just in order to do these different tasks. And so what scientists discovered is as soon as you start removing or modifying different parts of the enzyme, it starts to lose almost all of its activity. And so clearly this structure is a very complex structure that allows the enzyme to do what it does. And so the action of the enzyme is usually in the center of it. We call it the active site, and that's where the chemistry takes place. And working in the drug industry, what we try to do when you have, for example, some sort of phenomenon happening in the body that causes these enzymes to be overactive, what we try to do is to stop them from doing the work that they're doing by crafting molecular handcuffs. And so we craft these molecules. These molecules are to fit inside the enzyme and to essentially stop it from doing the work that it's doing. And so you can see, again, keep that analogy in mind because I'm going to be coming back to it later on. But the bigger question becomes, it's sort of when you see a a complex task being done. You know, a team playing hockey. We're in the middle of the playoffs right now. And you see all the things that are going around in order for this to be possible. And so these enzymes, there's thousands of them. There's all kinds of different types of molecules. All of these being regulated and functioning together in a complex choreography. What is the basis inside the cell that allows not just to explain the structures of all of these compounds, but also their interactions and all of the mechanisms that are taking place? And so essentially, what is the medium storing and communicating this genetic information? So earlier on, there's a fellow by the name of Mendel who did a lot of experimentation with peas. He had about 29,000 generation of peas, and he noticed that the different traits could be inherited, just like us. You know, we have, black hair, different colors of eyes, size, shapes. It's all part of the variation that we see in life. How is this variation? How are these traits inherited? So that was the question that a lot of scientists were wondering about earlier on. And so because proteins have 21 different amino acids, different letters, if you can say, they believe that that's where the information content was held, inside the protein. But they actually discovered that that's not the case. It is actually in nucleic acid, which we call, of course, now today, DNA, where essentially in this DNA, you only have 4 chemical letters, A, C, T, and G. But essentially in those chemical letters, all of the information, all of the genetic information, the genome, is contained. And so if you wonder, well, if this is the book of life, the book of instruction, the manual of life, how big is it? Well, there's, in one human cell, approximately 3 billion letters, so bits of information. That's a staggering amount of information. Just to try to grasp how much information this represents, if you were to write it out in a book, those letters that inside one of your cells, inside the nucleus, inside the DNA, we're getting really small, by the way. We talk about our gadgets, getting smaller and smaller. Well, this is ridiculously small, and the amount of information that it contains. So just to get an idea as to how much information this represents, it's sort of like if you were to take books, Times New Roman, and write each and every letter, it would take you 1,000 books of 500 pages each just to write out the genetic information in one human cell. So imagine walking through a library, 1,000 books, 500 pages each, and say, this is all the information that is inside one of my cells. Amazing. And if you were to take that genetic information, the DNA, and to take it out from one cell and stretch out that molecule, so just to see how long it's going to be, it would be about 1.7 meters long in one cell. And if you took all of the DNA from all of your cells and put it back to back, you would be able to go to the moon and back 6,000 times. Yes, because you have trillions of cells. So if you multiply that distance by trillions, that's how far you can go. All of that is inside your body right now. All of that genetic information. Absolutely astounding, mind-blowing. And so the question again is, well, this information, what is the language that exists in order for it to have meaning? Because any language, in order to have information that is useful, must share a code, a transmitter and a receiver that shares the same code in order for that information to make sense. And here's an interesting analogy. I told you that I would try to do a couple analogies to try to make these things easier to comprehend. When you and I communicate, when you and I do different things and we use information in order to do different things, there are 3 steps that take place. First of all, the thought that is in my mind. You cannot see the thought that is inside my mind. It's impossible. The only way for you to know what I'm thinking is for me to speak the word. And so there's the immaterial thought inside the mind, the head, but the material word is what essentially expresses the immaterial thought. And only once the word is spoken, the action can take place. So thought to word to action. And sometimes the action is simply understanding. And what we discover is that there's a very similar process taking place inside the cell, where you have the information contained inside the head of the cell, which is the nucleus, that is the DNA. But that DNA cannot leave the nucleus. It is stuck there. And in order for some of its information to eventually lead to an action, a word must be spoken, and that's the RNA. The RNA is expressed as a portion of DNA inside the nucleus, then leaves the head, just like the word leaves our heads, and essentially will go into the cytoplasm, where it will be translated into an action. And this is how information is being processed inside the cell. And the analogy continues. For example, if I say letter B, you know what I'm talking about, it's a letter B. But what type of information does this give you? Nothing. It's just a letter. There's no information. But if I say B-A-T, bat, so now you have a picture in your mind, a bat. So that is information. So letters by themselves contain no information, but the combination of letters according to a code, according to a language, now expresses information. You might wonder, well, what about the bat? What about the bat? The bat is broken. Capital letter at the beginning, period at the end, complete sentence. You see what I'm saying? So you have three levels. You have the letter, you have the word, and the words are put together in order to create a sentence. And essentially, genes are sentences. They are portions of a bigger book. And each of these sentences has a specific meaning and purpose inside the big book of life. You see, this is how it works. And so this is what we're talking about here. These 4 different chemical letters that are basically put together in the DNA. And what happens is that these are put together in 3-letter words, and these are essentially read one at a time to give you the different words, and these words are put together in the gene to give you essentially a sentence that has a complete meaning. And some of you, might have bad memories at this point, but just like we have grammar in our language, we actually have a grammar also in the genetic code. Oh yes. Essentially, all of these letters can be combined. Like, for example, UC will give you phenylalanine, you see? So you can have all different possible combinations of these 3-letter words to give you all the different 21 amino acids. And on top of it, you also get the capital letter, which is to say, this is where the reading starts. This is the capital letter. This is where the gene starts to be read. And you also have stop codons that basically say, this is the period, this is where it ends. So there's a powerful analogy, not just with the way that we understand and process information, but the way that it is being done inside the cell at the very cellular levels. And so as we take a step back and look at the big picture of what this involves at the level of the cell, remember, this black box that was not understood, that is now cracked open. We got a chance to see what's happening in these invisible cells. So remember, we are trillions of cells, not billions, but trillions of cells. But we all started from one single cell. That means that all of that genetic information that was there right from the beginning must be faithfully copied every single time that this cell divides to give us what we are today. And this has to be done without errors, because any mistakes are going to lead, in many cases, to diseases and all kinds of different problems, and sometimes even death. We know the problem with cancer often being caused by different types of mutational errors. And so how does this occur at the molecular level? Essentially what we have is all kinds of enzymes and proteins that are there that are going to unroll and uncoil the DNA in order to separate the 2 strands that are caught together and to being able to replicate 2 new DNA strands from the original DNA strand. And as I said, this has to be done without mistakes. And so there are even proofreading and repair mechanisms. So that if ever the wrong letter is being introduced in the new double strand of DNAs, there are mechanisms that are going to recognize the error, recruit the right enzymes and proteins, cut the connection, remove the wrong letter, bring the correct letter, put it back in, and stitch it up. Imagine a quality control mechanism that is inbuilt inside the cell in order to ensure proper expression of information. Absolutely mind-boggling. How does this information eventually lead to a word? And so essentially inside the nucleus, inside the head of the cell, you have all of the DNA, and when a specific gene needs to be transcribed, there's going to be proteins and different RNAs that are going to come, recognize the area that needs to be expressed. And imagine you're going through that library with all these books, and you got to find the right book and the right page and start at the right letter in order to find the exact sequence that is necessary in order to do the job. That all needs to be done. And so what happens is this DNA is unrolled, the sequence is transcribed into the RNA, so that that RNA then is able to leave the head of the cell, the nucleus, go into the cytoplasm, and eventually lead to the action that is necessary. And there's something here called splicing, which is a very complex and intriguing mechanism that we've discovered in the cell, which adds even more complexity to the process. Meaning that in order to get one single protein made, you might need a little bit of information from one book on one shelf, and then another bit of information on another book on another shelf, and these two things need to be brought together and spliced together in order to eventually have that exported from the nucleus of the cell to lead to the proper protein. So again, the levels of complexities are just astounding. And finally, the last step of this sequence is the translation of that information. So that RNA that leaves the nucleus, enters the cytoplasm, it gets read like a machine by, again, a series of different enzymes and proteins in order to find the right amino acids for each codon, for each word, stitch these up together into the right protein so that this protein will eventually lead to the correct action. And this is not the end of the process also. Even this being completed is just the beginning in some ways, because this protein is immature. It's sort of like a teen. It's got to mature. And so you're going to send it to this Golgi apparatus, where it's going to be folded in the correct three-dimensional structure. All kinds of pieces are going to be added to it. It's going to be then transported to the right place inside the cell, and at the right moment, in the right time, then the last transformations are going to be done for it to be activated at the right place and do the job that it's supposed to be done. I mean, this is what we're looking at when we're looking at the information content and expression of the cell and what a picture it generates. So let's take a step back here with the questions that we're asking today, which is the questions of origins. You know, you've heard about the chicken and the egg. Well, we got a different kind of question here. The question is, which one of these could have come first? Well, it can't really be DNA, because DNA is the origin for proteins and RNA, and DNA cannot be constructed or even copied without the help of proteins, so it can't be DNA. It can't be RNA, because RNA comes from DNA, and again cannot be transcribed without the help of a whole series of complex proteins and enzymes. And it can't be the proteins, because the proteins come from the DNA, they can't be made from without the RNA, and they need other kinds of proteins and enzymes in order to generate these proteins. So, you know, we talk about the chicken and the egg. We've got a much bigger problem here in terms of trying to come up with even some sort of suggestion as to how this system could have been generated. Evolution not only requires us to believe that the letters have appeared on their own, they require us for a code also to have appeared on its own, and all of the machinery which is interdependent between them to be there right from the start in order for this to be possible. We can't even construct a theory that would allow us to explain how this is possible. Okay? Some solution has been suggested here on 2 dimension by M.C. Escher. It doesn't work on 2 dimensions. It definitely does not work in 3-dimensional biological reality. Okay? What's interesting as we're looking at all of these different aspects of the complexity of life is we go back to Darwin himself, and he wrote something in this book that is very important, very interesting. He was a man of great integrity and great understanding in many ways. He said if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down. In other words, natural selection requires something that has an advantage, that has a function to be selected. Now, if there's some sort of function where all of the different parts that lead to this function cannot be gradually put together, you don't have a function. And therefore, natural selection has nothing to select. So the question that he's asking here is, are there systems in life at the level of the cell and higher organisms that cannot be gradually put together? That's essentially the question. And so this brings complexity, but a different kind of complexity. And this has been termed by many scientists, mostly Michael Behe, as irreducible complexity. What kind of complexity is this? Let's look at it together. A system, irreducibly complex, is composed of several interacting parts that contribute to the basic function, and where the removal of any one of the parts causes the system to be effectively cease functioning. You need all of these parts in order for to have function. Remove any one of these parts and you have no function. Remember, natural selection can only select something that is functional. Otherwise, it's going to go wayward. This type of system cannot be gradually produced by slight successive modifications of a precursor system, since any precursor is by definition non-functional. Okay? So the question is, are there systems in life that fit this category? Well, let me just first illustrate, because this is a lot of words, and sometimes a little picture helps. And so Michael Behe, in his book, uses the following analogy. It's a pretty simple one. He uses a mousetrap. And so the function of the mousetrap is to catch mice. And so a system that can catch mice must be able to accomplish that purpose. If it doesn't, it cannot be selected because it is non-functional. How many parts do you need in order for you to be able to catch mice? There are not many parts in a mouse trap. It's pretty simple. But the reality is, it is an irreducibly complex system. Because if you remove any one of these different parts, as simple as they might be, will you catch the mice? The answer is no, okay? So the question again is, do we find systems in the cell, in higher organisms, that fit this kind of challenge that Darwin himself has brought before us? And the answer, of course, is yes. I'll give you just two simple examples. Blood clotting. I mean, if you and I get cut, we don't need to run to the hospital. We should survive. We're going to be all right. And that's because there's a system inside our blood that causes the blood to clot and then therefore heal itself, and then we don't bleed to death. If you're a hemophiliac, that's a different story. Now you're in big trouble. And the reason is because blood clotting is a very complex cascade. There's so many things that have to happen in order for blood to start clotting. And you can imagine that's a good reason, because you don't want blood clotting to start for no reason, because that would be a terrible thing. We know what that does. So essentially, if you're just missing one of these factors, you get no blood clotting. And so something like this is not something that would be able to gradually evolve over time, because in order to have blood clotting, you need to have all of these things together. Here is the ATP synthase. This is a molecular machine that is found in every single cell. And essentially, this is the machine that allows the cell to use sugar to extract the energy, and then to use that energy to power its own biochemical processes. This is ATP. And so essentially what happens is you have this gradient that exists on each side of the membrane, and as that gradient equilibrates itself, it drives this molecular machine. And when scientists discovered the three-dimensional structure of the ATP synthase, they said it looks like an engine, like an internal combustion engine. It has a piston, it has a cylinder, it has a block, engine block, all of these things. And the reality is that once again, this fundamental machine that is essential to all cells requires all of these different parts that are like an engine in order for it to be able to pump out the ATP that is necessary to drive all of its biochemical pathways. And so we see once again that these types of systems are all over life. I just gave you two very simple examples here. And what's also interesting is that as we discover more and more about the different mechanisms inside the cell in the body, is we start to discover that a lot of our inventions that we're so proud of, we have patents on all our new discoveries. We discover that a lot of the principles that we've invented and that we've applied have already been invented and are already being applied inside our cells. Little did we know it. That's quite interesting. Okay. Now, beyond complexity, now we're going to move on to what I consider to be the most important portion of this specific talk. Okay? Is everything in the universe matter and energy? This is an expression that I hear all the time in scientific circles. Everything in the universe is matter and energy. Well, a lot of it is. A lot of it can be explained using that. But is that statement completely true? I don't think so. And I'll give you an analogy to explain why I don't think it's completely true. Let's say that we have a supercomputer, the world's most powerful computer. And then I download all of mankind's information on this computer. And I ask a team of scientists to come into the room and to analyze this computer to death. Everything that's possible in terms of matter and energy. Okay? So size, weight, what it's made from, temperature, you name it, what you want. Okay? And then I ask this team of scientists to leave the room. I close the door, I take a big magnet, and I go like this on the hard drive. And then I asked the same team of scientists to come back in terms of matter and energy, to do the same analysis. And then I asked them the question, in terms of matter and energy, is anything different? The answer is no. In terms of matter and energy, is anything missing? And the answer is no. And yet you and I know that something is missing. But it's not matter, nor is it energy. What is it? It's information. What is information? Information is neither matter nor energy. It is the organization of matter using energy. But here's the caveat, here's the catch, is that the fundamental laws of the universe are such that it's going to disorganize spontaneously information. Entropy, genetic entropy, okay? Yet the material laws lead to spontaneous disorganization. So the big question is, if everything is matter and energy only, what is information? And yet we know that life is built upon information, okay? And just like I started this presentation today by talking about how there are two broad categories of science, we discovered that there are two broad categories of information also. And the distinction between these is absolutely critical. So the first kind of complexity or order is called unspecified. And you have some beautiful examples here. You know, snowflakes and all the beautiful structures that we can see, crystals that can be formed. You know, these types of complexities are unspecified, meaning that they are the product of natural material laws. They happen spontaneously. They're spontaneous. And they also have a low information content. It is a physical phenomenon that is repeating itself to generate these structures. Yes, they're beautiful. Yes, they have a certain amount of complexity, but they have very little information content because it's just matter obeying its laws, generating all of these patterns and interesting forms and shapes. The second kind of information, second category, is what we would call specified, or complexity, I meant. Here's a motherboard of a computer. Now, the motherboard does not violate any fundamental law of the universe. In fact, we can explain its existence using all of the laws that regulate matter and energy. And yet, this type of information that we have here is not something that comes up spontaneously. It is not the product of material laws. It is not spontaneous, and it possesses a high information content. It is not repetitious. It is explained using the laws of the universe, but it is then exploiting those laws in order to achieve various types of function and processes. So the question now is, of course, what about life? In this slide here, you cannot make out what's on this slide. And it's on purpose, because there's way too much information. I mean, it would take us months, years, just to try to go through. This is not the motherboard of a computer. This is just a small part of the motherboard of life. What's inside the cell that allows it to do everything that it does. And you see these two posters here that I have on each side? This is these kinds of posters where you start to see all of the intricacies, all of the complexity, all of the structures, all of the information content that is being used in order to generate these structures and to regulate all of these mechanisms. Think about it. We would never suggest that something like this would arise on its own. But are we proposing that something like this is able to appear on its own? That, again, is a question that each and every one of us needs to answer. Okay? That's what we need to do. Remember also that when we are examining these different biochemical pathways and genetic pathways and all these things, there are dozens and dozens of people that have spent decades and millions of dollars just to identify sometimes one enzyme, one protein, one mechanism. So how much time, how much brain power, how much money has gone into making these things just for us to understand these things? That's just absolutely mind-boggling. And to say that we're spending all of this time and this energy to discover and understand just these little aspects, and yet we're spending all of this time and energy to discover something that appeared by blind, random chance? Is that reasonable? Is that reality? Is that really the outcome of all of these things? The mystery of the origin of information. Werner Gitt said, there is no known law of nature, no known process, no known sequence of events which can cause information to originate by itself in matter. That's interesting, because life, we said, is built upon information. Life is a riddle. We can't explain its origin. And now what we're discovering is that what life is built upon is also a riddle. We cannot explain the origin of information. And so we can kind of take Pasteur's biogenetic laws. Remember, only life begets life. But life is built upon information, so we can actually say only information can give rise to information. Information cannot arise spontaneously into a system unless you have previous information. The last part that we're going to look at today is now mutations, because this is a topic that we hear often about. And we're told that mutations are able to explain some of the changes that we discover in life. And that is true, absolutely, to a certain extent. But the real question becomes, are mutations sufficient to explain not just the transformation of life, but the origin of life? And so this is what we need to look at. So first of all, what are mutations? Mutations are alterations of pre-existing information. That's the first thing that has to be clear in our mind, is that mutations do not explain the origin of information. What they essentially do is they will muddle that information, they will change that information, and depending on how that's done, that's going to have different outcomes. It's sort of like a photocopier. If you take a photocopy and you photocopy and then you have a new photocopy and you take the photocopy and you keep, what's going to happen to that page eventually over time? The resolution gets worse and worse to a point where it's going to be completely black, okay? And so if it wasn't for the mechanisms that are out there that will protect the integrity of the copying of the information, you would be having all of this DNA damage, these mutations, this genetic entropy that would be creeping into our lives. And unfortunately, we are seeing this phenomenon to a certain extent. A lot of the diseases that are appearing are the result of mutations. But yet mutations, we're being told, are good in a sense, because it's through those mutations that new things can come out. And then natural selection will select those good new things and will allow it to drive the process of the evolution of life. That's the argument that's being heard. So let's look at this. It needs to be carefully considered. I worked at Merck for several years on ID, infectious diseases. And I worked on HCV, hepatitis C virus, and I worked on MRSA, methicillin-resistant Staphylococcus aureus. So sometimes viruses, sometimes bacteria. And essentially, I've learned a great deal of things that are all published, but that I didn't know about, and that actually very few scientists are really aware of, which is the mechanisms behind resistance. So let's look at this. So we've come up with all of these drugs that are able to target these different viruses or bacteria. And we're so proud of our chemical arsenal because we're able to help people heal faster. But lo and behold, we discover that we have viruses and bacteria that are laughing at our drugs, and they're just multiplying like crazy because they've become resistant. And so the idea is that, well, if they're able to do that on a short time scale, if you extrapolate over millions and millions of years, I mean, who knows what that's going to be able to provide, you know, this kind of positive evolution. But is it really positive evolution? Let's look at this. What we've discovered is that, as I said before, mutations of existing information and the mutations that have taken place in these different bacteria or viruses have changed the genetic information that's coding for genetic machinery, different metabolic enzymes, different membrane channels, etc., etc., etc. Remember my analogy earlier about the handcuff. I told you that I'd get back to it, right, with the enzyme. And so what happens is, let's say, I'll take an example of a drug, rifampin. Rifampin is a drug that would target an RNA polymerase. And so what happened is we made, chemical handcuff that stopped the polymerase from doing its job. And so by putting the drug, we stamp out that infection, and then the patient gets better. However, we discovered that now we have a strain that is able to live in the presence of my handcuffs and is laughing at it. It's fine. What happened? Well, here's my analogy. There was damage done by a mutation. That damage is causing the bacteria or the virus to being able to continue to reproduce, but there's a cost associated with it. There's a cost. It's sort of like if you have handcuffs, but I cut off your thumb, you can slip off the handcuff, but now you're missing a thumb. So you have an advantage over somebody who does have a thumb, but the disadvantage is that you don't have a thumb to do the other things that you're able to do. So what we've been discovering is that these mutations, number one, do not generate any new genetic information, damage the existing genetic information. Yes, it does give an advantage to these bacteria and viruses because of the artificial environment that we have created by putting these drugs in these people. But eventually, those mutants are not better. They're not superbugs. In fact, they're actually wimps. They're actually weaker because they're damaged. I'll give you another example. You need analogies for different concepts, difficult concepts. If I chop off your legs, you're going to have an advantage over other people. You say, what? Excuse me? Well, let's say you want to go at the mall and there's a big sale and you don't have legs and you come in a wheelchair, you can park right next to the entrance. You see? So you have an advantage in an artificial environment. It's not something that's overall good, but it's something that is going to be good only in a specific scenario. And this is what we're discovering with all of these mutants. Yes, they have an ability that the wild type, the wild strain do not have. But if you start analyzing them, they're actually weaker. They're just stronger in that artificial environment, like the mall environment, like the missing thumb environment. You see what I'm saying? And if you're not convinced yet, I hope that the following thing is going to convince you. I'm now going to show you an example of what would happen in a patient that has HIV. And you would give one single drug. No company gives single drugs now. It's usually 3 and usually 4 drugs at once to avoid exactly what I'm going to describe, okay? But what I'm going to show you is to demonstrate that these resistant strains are not more powerful. They're just different. So this is like, for example, the blood, the viral load that you would have in the blood of an HIV patient. So this is the wild type, and you have a couple mutants. This is just an illustration, okay? So what you would do is you put your drug in the patient, and you would drive down the viral load to a point where now, the patient is doing much better. But the problem is that what if one of these mutants has a mutation that allows it to be able to function despite being in the presence of the drug? What would happen? Well, you can probably guess that eventually resistance shows that this strain is able to survive in the presence of the drug, and it goes all the way up. Again, the question is this strain better? Is it stronger? Is it a superbug? Or is it actually weaker? Here's what would happen if you would remove the drug. So what we're going to do is remove the drug and see what's going to happen. And this is usually what happens over weeks and sometimes over months. The mutant eventually is going to be driven right back down to where it was pretty much before, and the wild type, the original one, is going to come all the way back up, which demonstrates that when they're competing in the same environment, that the wild type, the non-mutant, is actually more fit. And natural selection will select the one that is more fit. So these organisms are not super at all. They are damaged. There is no new information. The existing information is damaged. What happens is it allows them to survive in an artificially created environment. What about higher organisms? Well, there was an experiment that was done in the US over the course of many, many years where they took fruit flies, Drosophila, and they submitted these fruit flies to all kinds of different environments, all kinds of selective pressures, also in the presence of UV rays to really increase the amounts of mutations. And they did this for dozens of years, which is equivalent to about 80,000 generations, equivalent to millions of years of human, for higher species. And so the goal was to see what kind of evolution are we going to be able to speed up, to precipitate by us directing the environment, to see what we're going to come up. And essentially what would happen is that the new breeds, all kinds of new breeds, very interesting results, but what happened is they always remain fruit fly. And the conclusion from these experiments was that the fruit fly has already evolved as far as it can. Does it mean that the fruit fly has evolved as far as it can, or does it mean that our whole concept of evolution being, a fruit fly eventually being able to give rise to just about anything over time is flawed? That's the question, again, that we need to answer. Life displays a wide range of horizontal variation within kinds. Look at dogs, you have something like a Great Dane. It's almost like a small horse. Then you get a little Chihuahua that you can hold in your hand. I mean, they're both dogs, but I don't think they can even mate. I wouldn't want to see that. But the point is that there's an amazing amount of variation that's possible within kinds. And we see that. The adaptation, the flexibility, the plasticity of life that is inbuilt in the genetic information, in the genetic code. But we have never seen a vertical crossing between kinds. You know, the Chihuahua becoming a blue whale or an eagle or something of that nature over millions and millions of years. That is not seen anywhere. It has never been observed. And furthermore, it even defies theoretical explanations and model to try to come up with something that could explain how this would be possible. I finish now with this last two slides. You have here, little bird houses, somewhat complex. Bird houses made by someone. These bird houses cannot consume, they cannot excrete, they cannot fix themselves, they cannot reproduce, right? But we would never say that something like this can appear on its own. But look at the tree to which they are attached, from which they were built. Trees that are able to consume, that are able to excrete, that are able to fix themselves when they're broken, that are able to reproduce. And we would say that trees have appeared on its own. And what about us who took those trees, cut them down, and built these little houses? Where do we come from? Are we just the result of time and matter? With chance over millions and millions of years? Again, this is the question that each and every one of us needs to answer. This brings the principle of uniformity. Regular experience teaches us, this is what we know from regular experience, that only intelligent beings can design and construct complex structures that possess high-level function and purpose. And there is no greater example of all of these things than what we see in life. So what is our conclusion from life? Here's another image from M.C. Escher. If you look at this image, you'll see that the people on the outside are always going up, and the people on the inside are always going down. Doesn't really make sense now, does it? The theory of evolution is a scientific theory that has to be seriously considered. And when you look at it from a distance, it seems to being able to explain so many things. But when you start looking and breaking it down to the basics and looking at all the different aspects that are involved in the question of life, you start to discover that there's issues, that there's big issues when you start to focus and looking at the big picture, both of these things. And so what we've done today is essentially started by looking at the gap that exists between inanimate matter and animate matter, looking at the physical, chemical laws that exist, and is it possible to bridge that gap? And all of the evidence that we are accumulating are demonstrating that this gap is not getting narrower and narrower, but wider and wider as we understand more and more as to what life is all about. We then looked at the complexity that we see within the cell, the fundamental unit of life, the irreducible complexity, and how all of these different mechanisms are incredibly complex, and we cannot find even ways to imagine how these things could have appeared on their own. And then we have the information content, the information that is the basis upon which life is constructed. And just like we could say that only life begets life, only information is able to beget information. And so as we conclude now, we see that science is pointing. It is not speaking with specific words. Science is not telling us what the answer to our questions are, but it's basically pointing in a direction that is not the direction which wants to say that matter and energy on their own over time are able to give you the structures of life. It does require faith, as I said from the beginning, whether you're on one side of the debate or you're on the other. But the question is, again, what is the quantity and what is the quality of the evidence upon which we stand in order to believe what we believe? It requires faith to believe, but is it reasonable faith? And that's the question that we need to answer. So tomorrow what we are going to do, we are going to move on to the highest peaks on the shore of life, which is higher organisms. We're not just single cells, we're higher organisms that are made from cells. There's also a gap that exists, not just between inanimate matter and animate matter, but between the cell and higher organisms. What are there in terms of evidences to propose that we can come from single cells, or is this pointing to something else? Thank you very much.